Héros, héros. Est-ce que ça bouge? C'est bon. Ok.
regarder notre reste. 我也可以放吗？因为我们就是有做 live Facebook， 所以我们就希望观众在网上就可以听到你的声音。
Yeah, we are one of the speakers. Yeah. Thank you. So I see. Exhibition space for people to attend. So everything's become very flexible. Otherwise, this kind of rooms wouldn't be possible because they use power companies. It's actually power companies to release the whole building. Yeah. I know. Tea. Still quite. I thought you wanted the lady calling in. I thought you wanted to speak to her about something. Godfather said it was very relevant. Later? I'll just have a tea. I can do it. Yeah, it's very
like
hard
close
to
my
find.
house, to my workplace.
It's quite hard to find good bread in Taipei. I mean, usually most of the bread are the Japanese style ones, so it's very soft.
There are some bakeries too, like in Tsai.
Yeah, well, I live in Academia Sinica. It's very far, you know.
Yeah, but Academia Sinica is from everything.
Yeah, for the centralized real city people, Academia Sinica is like.
But it's not that far, but it's not really well connected.
It is.
Public transportation.
So it's actually a historical fact. So when the MRT was built, they were thinking whether we should have a station. At that time, they had the final station at Academia Sinica. Obviously, it would be quite nice. The president at that time asked some researcher from the Institute of Economics, and they worked out the cost of building a station next to Academia Sinica will equate to giving every family household a Toyota, a car. Yeah. Because at that time it's all residential, right? I mean, thinking about Nangang 20-something years ago is very different to now. Yeah, and so they say, and also for another reason, because that would be the final station. So a lot of people from outside Taipei, they might come in the morning, park the car in Academia Sinica, and take the station to center of Taipei City, so they can walk. And so they probably, one of the reasons is that they don't want Academia Sinica to end up being a big car park of, the other companies. It did make sense. I mean, at that time. But I mean, for me, it would be quite convenient if there's a station nearby. But it's okay. I mean.
But there's a bus.
Yeah, the bus is quite nice, but Nangang is also the area where it rains a lot. So they are thinking to build all these, the sky bridges that we walked in around the Taipei 101. So this is their plan, I mean, to kind of connect everything around that area. I think it would be nice. I mean, yeah, and they actually plan a bicycle kind of sky bridge thing from there to Academia. That would be nice for me. I mean, that would be just for me, would be perfect. But I will be probably retired by the time it's over. There's a canal. It's very hard to find a good canal in Taiwan. I went to Paris, and next to the hotel is the bakery. The canal is so good. Yeah. I really like it.
You put manually
Yeah.
so when we had
It's already inside.
but this is made of what? This bubble.
Tapioca. style is ready for.
Yes, yes. Just need 10 minutes to adjust some layers. So I just tried, it was the first time I put it together, so
But you gave a kind of practice in NHI. Was it the same?
it was the same talk. So, but yeah, it went well actually. I thought it was going to be a bit more round. But I just need to
Yeah, yeah.
that we had the first day.
I don't think so. It looks like a Japanese brand. Maybe I'm not. But either way, she's it will be
That fermentation.
yeah.
All the best of
Yeah. Some water. When I was working in McDonald's in Nottingham, I usually work in the night shift. I don't know if you still remember Cine World. You know, there's a cinema in Nottingham.
What's it called?
Cine World. Cine World. Yeah, but anyway, it's the one close to Duncan. We usually have our last orders before Sunday, before closing. and everything after this. And when we close the restaurant, we need to throw everything out. We cannot give to the members. Yeah. Even though they are considered wasteful, it will be even more of a trouble if we gave to someone and they have some food poisoning. It will be even more responsible for McDonald's. But I always, me and my friend, we always just overcook 15 nuggets. Just before fry the 15 nuggets before. Then, it's close. It's wasteful. But then us, we can actually take it. Yeah. So we take like 15 nuggets and fries, and we just go to the cinema to watch the last movies. And this is usually our typical weekend, which is quite nice.
Yeah.
But yeah, maybe when we were young, imagine all the calories.
At the end of the day.
I know. And it's just a bag of fries, bag of nuggets, and a bag of sweet and sour sauce.
There is a nice cinema in the
Broadway, yes, Broadway. Yeah, it's nice. I don't know if you remember, there's a bar called Picture Piano. It's in the church. Yeah, I think it's quite nice. And the Jerusalem. When my friend comes, we usually go to Jerusalem. That's supposed to be the old. It's okay. That one is okay. Picture Piano is very nice, but weekend is not so nice because the long is okay. Yes.
And she comes back and
Yeah. Thank you. Oh, okay. You want to sit also in the front when I'm presenting, or you can see the yeah. I think it's
in the back. Next to the booth.
Yeah. Sounds good.
Also, this is dangerous, yeah.
Yeah, I know.
Be careful. Yeah, we'll see
Yeah, thank you.
of the Science Bubble Tea series. So the Science Bubble Tea is a flagship event from the French office in Taipei. So my name is Pauline. I'm the attaché for science and technology at the French office in Taipei. And so tonight we have the pleasure to welcome for this Science Bubble Tea two great scientists. So Gianni Liti from CNRS and Jason from Academia Sinica. And this is a great honor for us because both of them have been laureates a few months ago of the iconic France-Taiwan Scientific Prize. So this is the most prestigious scientific prize for French and Taiwan researchers developing outstanding research together. So I had the occasion to attend the ceremony at the Académie des Sciences a few months ago and to hear the great lecture you gave there for academicians. And so tonight this is for you, our public that Gianni and Jason will present their research. So it's a very groundbreaking research on yeast and genomics. So we all know a bit about yeast, okay, that yeast is needed to make bread, to make wine, to make beer, okay? But sometimes we don't know much further about yeast. And genomics is a bit mysterious for all of us. We know it's about genes, and this is very important. So now we want to hear more and to discover with you how with yeast we can understand more about population genomics. So I would leave you the floor.
Yep.
Thanks again for joining us tonight. And then my colleague Clémence, who is a great organizer of the Science Bubble Tea series, will moderate the Q&A.
Yeah, thank you. So thank you very much for this kind invitation. It's a great honor to be here with my friend and colleague Jason Tsai. So I think Jason will go first with his talk.
Yes, we will go first with your talk, then the second talk, and then we will. So I will ask you some questions, and then we will open the floor to the public. So if you think about questions, note them, and you will have the opportunity to ask them at the end. Okay, so we can start with the first presentation.
Okay, thank you. So I'm Jason, and it's very nice to see you all. I'd like to thank Pauline for the very kind introduction and the invitation. So I work on yeast, and I kind of start from the evolutionary scenario, right? So this is, most of you, when you talk about evolution, you maybe think about the Charles Darwin, and he gave a very famous book on the origin of species. And in his notebook, he tried to have this, but I think, statement of, how do you recapitulate the relationship between species? And this is usually represented by a tree structure. So if you have a species B, C, and D, you can recapitulate their relationship based on the distance of the tree. So B and C are probably more closely related compared to D, and then the furthest being A species. So, and if you talk about microorganisms or microbes, then obviously you have the great Louis Pasteur. And actually most of you probably would know that he actually started his research about fermentation. And then he started revealing that all the processes are probably biological and the discovery of microorganisms. And just a very interesting thing that the two great scientists actually overlapped in time. But probably because some French-British rivalries at that time, the two probably didn't interact with each other that much. But unlike today, Gianni and I had a lot of interactions. So, and this is the famous quote that I like, there's more wheat at the bottom of these 100 liters than in all of the philosophy books in the world. Okay, so when you have the two, you have essentially, we want to talk about evolution and microorganisms. And this is kind of the core of my research. So this is a typical setup of my lab, which consists of different parts of a setup. You go to a forest or you go to some sampling site, you do some experiments in the lab, you do some analysis, and probably you need a lot of coffee for thinking. So kind of a quick summary. Taiwan is actually an island with quite a vast amount of biodiversity. And this really allows us to study some of the fascinating organisms, right? So this is an iconic species, the fungi that actually glows in the dark. And we were able, because it's an island, we were able to work across oceans. We work with some of the very new organisms. So this is a case of salamanders, and this is actually has been one of my favorites. So there are actually hydrothermal vents around northern Taiwan, like Guishan Island, and they have this particular hydrothermal vent crab that actually can live in a very, very, very high temperature, around 80 to 90 Celsius, and still quite nice, thriving. And I always make a joke, they are not actually cooked, but they are, very, very thriving over there. And you do have a lot of very nice systems to work in Taiwan. So for the purpose of today, I work on microbes, but especially yeast, which is the kind of main player for today. And just a very quick introduction, microbes are everywhere, including yeast. And they are practically found in every environment, and they have a lot of functions involved with host and the environment. So how does the yeast actually contribute to all this health, food, and energy? And probably you can think of is you have a particular substrate and it does something, and you have the products that are very, very related to human civilizations, right? And today's main focus, or Gianni and my focus, has been this particular organism, Saccharomyces cerevisiae, right? So it is a yeast, and it's arguably one of the most human-utilized microorganisms, and it is a model organism in research, right? So many of the fundamental principles of biology were discovered in this species, but little is known about its ecology outside the lab or outside the human environment. So we have this particular challenge or practical challenge. We have a lot of yeast that we use, or the commercial strains we use for bread, butter, and biotech maybe. But what are we missing? In terms of the wild diversity of yeast, if there's any. Right, and so my lab's motivation is we try to isolate these microorganisms from their natural environment, and hopefully we can bring them back to the laboratories and study them, as if, and trying to understand in their natural habitat, what do they actually do, what do they actually contribute. But where is the natural habitat of Cerevisiae? Right? And again, usually you tend to relate this organism with human civilizations. Bread, beer, alcohol, and it really goes back to many years, right? This is a Japanese sake making process, and it really goes back to 6,000 or 7,000 BC. So this is a typical. This is actually a recipe to how to make a beer back in the days of around 6,000 BC, and people actually replicate that, and it doesn't taste very nice. And this is the 7,000 BC, where you scrapped the innings of these jars, and you were able to isolate live yeast cells, and then revealing actually the usage were to do with fermentation. Okay, so we do have a map of how the yeast goes around to different places of the world. And this is actually a field that's established through my colleague and friend here, Gianni, right? So you have a lot of things, particular events of how the yeast travel around the world. And for me, I'm interested in a sense, how they come about before we humans start to use them, right? So this is kind of a thing that got me interested. And so where are the yeast? There were many documentations that they were isolated in the forest, these species, and the oaks and the wasps. And particularly, they are the ones, the common species that were found in the broadleaf forest around Taiwan. You know, usually from 1,000 meters to 3,000 meters. And this is a typical sceneries of the broadleaf forest we can see in Taiwan. And so kind of the picture would be, you know, can we understand where they live, you know, how do they interact, and finally, what's their evolution? Okay, so this is a typical scene of the broadleaf forest, if it's not with the hiking trail, typical hiking trails, right? So this is a typical environment of the Taiwanese broadleaf forest, you know, where as you can see, and there's a lot of Fagacia tree species. This is one of them, and these are many dominant Fagacia dominant forests. Right. Okay, so we have been sampling since 2016, right? Basically, going collect all the different substrates of the forest, and we put them, take to the lab. We try to incubate them with some sugars, you know, particular recipes, and then after a few days, you see bubbles, right, which are signs of fermentation, and you see kind of a pellet, which suggests there are microorganisms inside. And we streak them out. You know, we try to, you know, see if we found anything, and indeed, you know, we did find a lot. Otherwise, I wouldn't be here today. But so we had a total of 100-something isolates of these species. Think of it as different individuals of these species, right? And they're actually all over Taiwan, you know, in different parts of the forest. And one of the questions would be, how genetically related are these isolates to the rest of the world? There are a few hypotheses, and we plot the phylogenetic tree, right? So the important thing is, this is a tree that's based on representative isolates around the world. And the green branches, as you can see, represent the Taiwanese branches. And quite interestingly, it also occurs to different places of the tree, right? So it's not just one particular place where all the Taiwanese isolates look the same, right? It's actually all the world isolates. You do find them in Taiwan, in that particular sense, right? And if you actually put it on the map, it looks like this. So every color represents a particular lineage or population. And you have a very high number of populations or genetically different lineages as opposed to the rest of the world. And this actually is consistent that, you know, if you follow some of the human genetic stories, usually the region that has the highest geographical, that has the highest diversities usually represent some of the geographical origins, right? So this is kind of consistent with, you know, the yeast origins comes from East Asia. And again, there may be a few scenarios as well, right? So, you know, why Taiwan has so much yeast diversity? Maybe they arrive from China, and all of a sudden the environment actually makes it highly mutable. Then you become many lineages. Or actually, the actual yeast species originates from Taiwan and spread out of the world. This is less likely, but very appealing to maybe some of our politicians. But probably a more realistic to the scenario is they have come to Taiwan through multiple time events. And because the environment has been quite stable in Taiwan, they kind of returned and stayed here for many, many years, right? Okay, so still a lot of outstanding questions. And actually, the next big question for us is, what about the rest of Asia? Southeast Asia, they have also a lot of forest and also very, very different environment. What do the yeast look like in these places, right? So we went on sampling, and this is a typical forest in Vietnam. Actually, it looks the same if I told you this is from Taiwan. And this is a typical sampling trip that, you just have unexpected rainfall. This is before my student sent me a text message asking, does he have any travel insurance? And I replied, yes, so he crossed the river. But yes, we do have a lot of isolates when we started to think about the next question, right? So again, with my friend Gianni here, we were thinking about, how does all these microorganisms move and evolve around the world? If the highest diversity were indeed in Asia, is this consistent with all other species and other microbes? Okay, but it doesn't stop there. As I mentioned, you can take the species back to the lab, right? And you can start asking some questions about this species, And of course, you can, with all these yeast species, you can actually create home brews in the lab, right? So this is a typical lab exercise, where we take our wild strains and we brew different batches, and the students will have a blind test to see which ones were the best. And we can actually do some systematic compound testing to check, with this very little micro fermentation takes place, and we want to show if there's a particular strain that has a particular compound that makes the alcohol taste nicer, right? Maybe this is more of an application viewpoint. So on that note, I'd like to kind of conclude, our ancestors have been using, going from the environment to take something, and then we start using it, and yeast has been one of a perfect example. But now we are, in a sense, we are just trying to do the same, but in a more scientific way. And yeah, that's kind of the end of my talk. And just like to acknowledge my lab members and all the collaborators, including Gianni here. And thank you very much.
Thank you for your presentation. So we can put the second one. Gianni, it's your turn.
Oh, sorry.
Okay. So again, thank you very much for setting this up. It's a great pleasure to be here with Jason. So Jason said pretty much almost everything. So I will try not to overlap too much. This is my team in Nice. So we are part of the Université Côte d'Azur, and we are in the Center of Cancer and Aging Institute of Nice, which also is funded by CNRS, INSERM, and the CHU of Nice. We do actually work also in some projects are more related to cancer and aging. So, and I will tell you a little bit on on how great is the Saccharomyces yeast as a model, not just to produce goods for the human society, but also for study essentially any aspects of biology. But today I'm going to continue what has been introduced by Jason on how sort of these Saccharomyces variation vary across the world. We heard about the Asian origin, the Taiwanese divergence of the species. So what about the rest of the world? I'm going to show you a little bit today. So as Jason says, so there are two aspects of yeast that make a very interesting microbe. So the first one is this sort of human interaction. So actually this was taken from the National Palace Museum here in Taipei, and it's a wine jar from approximately 4,000 years ago. This is one of the examples on how long humans have been interacting with yeast. And as has happened for many plants and animals, actually this has led to sort of what is called this process of domestication. So basically human has shaped the biology of this species and select, in this case perhaps involuntary, select for specific traits. But the other aspect of what I was mentioning before, it's actually Saccharomyces cerevisiae has become, over the years, one of the most important biological model systems. And this actually dates back almost 100 years ago. So this was, there were some image from 1937 where this person, Ovid Winch, he was dissected, he was plating cells of yeast into an agar place, tried to select for different phenotype. And this was done in the Carlsberg laboratory, so it's in Copenhagen, and it's regarded as the foundation of yeast genetics. And from here, there are more than 1,000 labs across the world that use barley yeast as a model in biology. And basically, these two aspects of fermentation and studying biology, they came together around this time. So, and this actually is one of the strengths of this model system. So, my team in NIS, we use mostly population genomics. So we sequence genome of yeast that are isolated across the world. And essentially the genomes, it does contain information that can be used to infer the evolution of the species. It can allow you to travel back on time and try to infer what has happened to this species. And the other things we do it is actually we use this information to try to understand phenotypes, so to understand how this variation that we observe, for example, a strain that is good to make wine or to make a beer, how it does sort of can be connected to its genetic variation. And one of the strengths of the yeast actually is because it's being used as a model system, you can control its biology very well. So for example, here you have single cells, so we can analyze single cell of yeast, and we can color them in different flavors. We can induce sex. Actually, yeast can go, like animals can go through sex, can make male and female gametes. These are called A and alpha, and we can control also the sexual cycle. We can make clonal expansion, so this would be like a tumor expansion. We can make a clonal expansion of a yeast of millions of cells. And this actually has enabled biologists to study these organisms very well. So Jason has shown this phylogenetic tree. So this is a way to represent species, how closely are related to each other. And one additional species we've been studying over the years is called Saccharomyces paradoxus. So this is the closest known species to Saccharomyces cerevisiae. Is considered to have been split to cerevisiae about 3 to 5 million years ago. So this was before probably humans started to play with it. but he has this characteristic of not being associated with humans. So this to us is a nice parallel to understand the human impact on the biology of the species, on the genome of a species. And actually these two species, they belong to even, I don't know, bigger family. Again, you see this is a tree, and the more closely related, the more closer in this tree and the more these species are. For example, Micata, Eure, these are all Saccharomyces species. They all have this property of fermenting sugar and basically have a different degree of divergence. But actually they can still come together and they can make hybrids. And perhaps the most iconic hybrids that has spontaneously arise over the years is what is called Saccharomyces pastorianus. So this actually is a cross. So there's been an outbreeding between Saccharomyces cerevisiae and this other species here that is called Saccharomyces eubayanus. And basically this hybrid here is what is used worldwide to make lager beer. So this is one of the style of beer fermentation, which will be what you have here in Taiwan, called Taiwan beer. So I go directly here. So at some point we sequence, thousands of these Saccharomyces cerevisiae, mostly asking through colleagues. And in this specific study, with a colleague Joseph Schacheren in Strasbourg, we sequenced 1,011 Saccharomyces cerevisiae strains called the 1002 Yeast Genome Project. And basically we have analyzed their genomes, so these were fully genome sequenced. And this is the tree that we obtained. So you see there are these very long branch, so this means these strains are very different, and this very long branch is what we call at that time the Taiwanese lineage. So this was highly diverse lineage. There is more than double genetic variation, so these are twice more different than any strains that come from the rest of the world. So you see also these green and red circles. So this indicates the origin of the strains. So for the green, these are all strains that come from the wild environment, whereas the red strains here, they mostly come from domestic environments. So for example, here we have strains coming from wine, from olive oil, from French cheese, and other fermented food and beverage. And here instead we have many of these wild isolates that they come from Asia, actually, from different parts of Asia or North America. There are two exceptions. One is these green strains here, which I'll show you a little bit later if I have time. These are actually strains come from European forests. So this is what we call the Mediterranean oak clade. And these are wild strains. And here instead is this red dot here is strains are used in Japan to make sake. And basically these are independent domestication to the West lineage, showing that actually human has independently domesticated multiple times for different products by the yeast. So let's take a little tour around the world. So here, these are the wine strains. So this would be this bottom left group here. There are many strains. We have like 300 strains. And we can even see subgroup within these wine strains. So what do we have here? So we have, for example, strains from Georgia. So Georgia is a very specific way to ferment wine in this terracotta amphorae. And Georgia is considered to be one of the birthplaces of wine fermentation. Then we have strains that are used worldwide to make wine. So if you actually go to France, Italy, but also if you go to Australia, you go to Chile, you go to South Africa, and you sequence a strain for making wine, most likely will map into this clade. So this actually highlights how human has been moving these strains in the domestic environment around the globe. and basically has changed in a way how the species is distributed geographically. Also, sequence strains were collected more than 100 years ago. This is a yeast collection present in Italy, where actually I did my PhD, and we sequence strains were collected from 1920 to 1960. So this was before actually humans start to add yeast to the wine fermentation. So these were more spontaneous fermentation. They still map here, and there is strains that are actually used as probiotic. If we keep going around the tree, so these are actually strains were isolated from stool of healthy people in the French Guiana. This is probably due to the fact that they drink a fermented beverage that is not pasteurized, so, and probably these strains are resistant to go through the human gut. And instead, this group of strains, they are isolated from people that are infected by other organisms like Candida, or they are coming from people that have a poor immunity system, so they are immunocompromised people. And in here, Saccharomyces cerevisiae can become a pathogen. So most of the time it's a good friend, but in different conditions it can become dangerous. Perhaps one of the most interesting clades is this one. So this one is actually strains isolated in Malaysia. So this is Bertam palm. These actually, this flower, they have a nectar that is spontaneously fermented by Saccharomyces cerevisiae, and it's drunk, this nectar, this fermented nectar, so it's the same alcoholic fermentation of a beer is drunk by this little primate, and these strains have a very peculiar genome organization. And to conclude the round, so this is again this Asian origin with Taiwan as a sort of a center of variation and origin of the species. Just to conclude, I don't know, many minutes. So we've been looking at the origin of the species. So this dates back a few million years ago. We've been looking at the origin as Jason nicely showed before, and this can be sort of time to this kind of time frame, 680,000 years ago. We infer a sort of one single out of Asia event that has funded the rest of the world population. So this has basically resulted in establishing natural population, especially in the North Hemisphere, but more occasionally like the Malaysian War, or in Ecuador you can find this white lineage. And more recently, actually, what we've been studying is the more recent history of the species. So this is basically studying this natural population in outside Asia. So this, for us, focus has been Europe, and whether we can better track the domestication, for example, of the wine strain. To conclude this, so we actually been starting this sort of continental sampling in Europe, like Jason has shown in Asia. We're actually collaborating with Jason. We put together a group of other scientists that were interested in help us to collect samples. We didn't have travel insurance, so we have to get a sample in another way. And we sample all these sites across Europe. And now we have a new collection of yeast isolates of different Saccharomyces species that are been isolated across all Europe. I skip all this, and maybe we'll get back during the questions to this. Just to like to thank the team that is listed here, the current team, all the collaborators and the funders. And it's a group picture, so actually when we think about Nice, we always think about a city next to the sea. Actually, we have a nice mountain, about 90 kilometers, where we can go to ski for the day. There was one of the lunch break during the ski day. Thank you very much.
much for your presentation. Yeah, you can sit for the Q&A. So I'll start by asking you one question. So just before you were talking about the samples, the fact that you take samples in both regions, and I'm wondering if you could develop and explain what are your complementarities between your two teams, and what's the specific focus of your collaboration?
Should I? Yeah. Yeah, actually that's a great question, and it goes one of the central topic of science and maybe challenge is about reproducibility and how much we can compare, for example, what I do with what Jason does. So one thing to tackle that is actually to have a protocol. So we have a very defined set of steps that we do when we do both the collection, but also when we process the samples to try to isolate our strains. And it's true, actually, this is very challenging because everyone also has got this specificity. Of course, the samples themselves will be different. So we have adapted the follow-up protocol to the specificity of, for example, of the place where we do it. But overall, we try to be as consistent as possible, or explore each other's protocol, and then try to combine things together. So it's very often, for example, me and Jason will exchange the genomic data, because that's also is another important part of comparing things. And we'll run each other analysis, because the way you analyze data will be different between labs. So we try really to make things as homogeneous possible to compare.
Do you want to add something?
No, I think everything is good, yeah, as Gianni said.
So I have another question. So you've talked about the fact that your laboratory is also working on aging, cancer, and also in your PowerPoint you have a mention of health. So could you explain how your research can be applied to medicine, if it can be, and if you have concrete examples on that?
Yeah, I think so. More from the fundamental scientific point. I mean, yeast has revealed probably many aspects of biology. If we consider both molecular biology, genetics, and cell biology, there's been a number of Nobel Prize awarded to yeast researchers, for example, that have unveiled how autophagy work, or how the cell cycle of a cell work, or for example, how telomeres, or the end of chromosome work. So this already is very important, because often we learn things, or we used to learn things first in yeast, and then we look at other eukaryotes, including us. For aging, there's been different models that have been developed in aging. Aging in a single cell eukaryotes is different from human aging. But again, many of the processes that occur towards cells are conserved from yeast to human, like the genetic regulation. So, for example, one aspect of extending lifespan is to decrease nutrients. So this is called caloric restriction. And this works from yeast to flies to worms to human. More directly, we'll be using yeast to study how drug resistance evolves. So this is an important health aspect. For example, if we think the problem of antibiotic resistance, but also for the case of chemotherapy. So one of the problems is clone acquire new mutation and become resistant. So we can extrapolate information that we learn in yeast on how things, for example, cancer could progress and develop.
For my part of research, I've used to work on a bit on the pathogenic yeast now, but that's kind of a side project. So basically, is to understand how pathogens infect humans, right? So that's usually a big aspect biomedical research. And another part is probably not directly contributing to human health, but maybe the life around it. So in essence, a lot of these microorganisms have been used on different things. So like food industry, so you can be thinking about in a practical sense how to make a better beer, alcohol in that sense, or in a more biotech point of view, how to make more efficient and clean fuels. So for instance, one of the side projects we are currently conducting would be, we know the model Saccharomyces cerevisiae can be engineered to produce such and such fuels, for example, isobutanol. So what happens if we go back to very square one? If we have identified a wild species that can have already produced a lot of isobutanol from the start, and then we can have a good foundation along with some of the engineering technique we learned from the model yeast to further engineer this particular wild yeast to produce even more isobutanol. So this is the kind of concept we have been going around for quite some time. And again, it's going back to what our ancestors have been doing already for thousands of years, taking what's already present and have evolved for many thousands of years. But based on scientific knowledge, we try to optimize it.
Thank you so much for these very interesting answers. So now we will open the Q&A. So what's the first question?
Okay, thank you for the presentation. My first question is about the population genomics. There is at least 2 locations missing. I was expecting missing. It's the Moon and Mars. I'm wondering if how
the Moon. Sorry, what did which location is missing there?
Moon and Mars.
Okay.
And the question is like, how useful is yeast in deep space exploration? How can it really help us survive this kind of environment?
Yeah. Actually, it's a great question. So scientists have included microbes and also yeast in sort of spaceship experiments for many years. So they look at the effect, for example, of radiation or microgravity and things like that. So I think so far the main investigation was to look at what would be the effect of this condition to the cell, to the genome. So whether they will accumulate mutation. This could be again interesting also for the person that fly up and to what they are exposed in terms of conditions. But more recently, actually, they came up also with a new device. So yeast are used also in biotech to produce protein, which they could be also used to produce food supplement and so on. So one idea of going up is also to have a sustained population that doesn't need to travel back and so on. So actually they're thinking about producing goods and food and so on directly in the space. And these are actually where people and scientists are exploring at the moment. So there are already experiments doing that. So to see how can be transferable this type of technology from there to other planets.
But do we know if it's successful or not? We don't know yet.
No, I think they're really still in the sort of proposal stage, these things. And these last things I've seen it was to put things in the European Space Agency. So it was not in the planet, it was in
Yeah. Yeah, I guess I would probably give similar answers. So a lot of the research is being, or at least the ones you can try to replicate it on Earth, would be to have create an environment that has very low gravity, right, or nearly almost no gravity, right, and see how the cells grow. And Gianni also mentioned basically if you have yeast working as cell factories, right, if you have a self-contained cell factory in a space station, then you would have alleviate some of the potential health deficiencies of the food intake by the astronauts. And that's actually very, very important, right? So I think you are probably, when you ask this question, you are probably referring to how the actual yeast adapt to the space environment. Personally, I'm kind of an ecologist by training, so I would love to go to space and then actually work on the yeast species there. So maybe that's something the French government can think about in the next couple of years. Yeah.
Yeah.
I think that's a question for you.
Sorry?
I think that's
Is it about the French government? Maybe for Paulino.
So there was a second question.
Thank you for that great talk. So
He's our friend, Mike. That's why he's saying okay.
this wasn't a planned question.
It's all fixed.
So you showed those pictures of the old jars from like 6,000 years ago or something.
Yeah.
So are we dependent on just finding jars to know how long ago we domesticated yeast? Or can we use the DNA sequence data to get an idea of how long we've been in partnership?
Okay, I guess I can take a stab at this question. So I'm very interested in this. So there's a whole, I would say, wild and exciting field called experimental archaeology, right? So you basically take relics or antiques and you try to figure out, what's the usage of this particular apparatus. So for example, if you see the vase or the jars, you don't actually know if it's actually used for burial purposes, right? You only put it in when someone is dead, or it's actually used for vase, or it's actually used for fermentation purposes, because they all probably look the same, unless there are historical records to document the usage of these jars, which are really lacking. So a lot of sequencing. So in terms of that particular example, they actually isolate the innings of the jar, which there are still some spores which were kind of dried out but revived when the scientists actually try to isolate the scrappings of this. But it, as you said, it does rely. I think it does rely on the particular environment that allows the preservation of these cells to stay for a long time. And often it's a bit like the ancient DNA research. You need good specimens to find out about this. And so there's also actually a field of ancient DNA which could include the diet of some of the ancient humans, they were taking, and there were some studies of yeast cells. So that have actually confirmed, yeah, of course, there are yeast cells in the human gut in some of the ancient humans. But other than that, I think it's a very, very exciting, a very, very cool field. But it does take a lot of experimental challenges to process these samples, as well as sometimes just being fortunate that the apparatus is in perfect preservation conditions for the cells to be viable. I remember when I read the article, I was very, very excited I went to the Institute of History in Academia Sinica, and I met a professor who was in charge with all the ancient burial sites in Taiwan. There were actually a couple of these in various parts of Taiwan, and I was like, oh, could you just give me a apparatus for me to work on? And so he gave me a piece of jar relics, so that it's a part of a jar, but because everything is so precious, he only gave me like one piece. So and obviously for me it was later turned out to be a failed experiment because I just couldn't find any these cells. So it does depend on a bit of a chance. Maybe Gianni has better luck.
Yeah, I think also we haven't been lucky with finding good specimens. I took a sample with a colleague in Paris at the Institut Jacques Monod, and it was aware at the Louvre Museum there is a piece of mummified bread that would have been nice things to also try to sequence for ancient DNA, but we never managed to get there. But yeah, I think there are these come with lots of challenges. Ancient DNA studies, works nicely for human animals and so on, but microbes also exposed to contamination with nowadays. There are things like lake sediments and so on that have been sequenced, and this may be more interesting to look at longer evolution time scale before, let's say, domestication, and see when disease that, for example, come to Europe and so on. But yeah, we don't have made any progress on that yet. Thank you.
Okay, thank you. Are there other questions in the room?
Good night. I have a question about, is there worries about the biodiversity decreasing under climate change in the yeast field? Because in the agriculture field, a lot of scientists are worried about the climate change might cause some crops that cannot survive under future scenario. And for the yeast, because both of your study mentioned that there are some species are collected in forests or some fields. This might under threat of the environment change or some landscape change. So maybe some species might extinction. So I just wondering, is there some preservation ways to keep the diversity of the yeast population or not? Because for the agricultural field, people will establish something like a seed bank to preserve the crop diversity. Is there also some ways similar like these to preserve the yeast diversity?
Okay. So that is a very good question. So I think in terms of a microbe conservation as a whole, or whether this is yeast or bacteria, I think we are still in the discovery phase. In a sense, we are still trying to document how many species of yeast are present in the forest or in any kinds of environment. And so it runs into a conundrum that, at the same time, there's climate change, there's, global warming and deforestation, but at the same time, we are still also at a phase of trying to discover how many of these species are present. And, at least from my point of view, yes, I think they are very, very important. And obviously, practically, we haven't captured enough diversity in terms of yeast from any environment so far. But it is very difficult to talk about conservation because we do not know which species we need to conserve yet, right? Because we are still in the discovery phase. At least that's still kind of my viewpoint. And and the second question would be, even if we document all the species, all the yeast species, say, for instance, this particular forest, then choosing which species to conserve and preserve is another question, right? I mean, a lot of the conservation sciences is moving away. It has really transformed rapidly in the last 10 or 20 years, thanks to the sequencing and some of the monitoring program. But again, conservation really stems from the human side of things. You choose to conserve, I don't know, pandas when you, when maybe based on, particular trait of this particular creature. But whereas some creatures are not to be considered a priority simply because of, it doesn't probably have a desirable trait. So in that sense, it's very difficult to choose a particular target objectively, and it does relies on everyone to establish this. But for us, at least in my lab, we are actively documenting some of this diversity. And for instance, if there's anything that's going on in maybe deforestation in Taiwan, for instance, I can just send all my collections to Gianni, right? So that part will be safe. But this is kind of little things we can do right now.
Yeah, maybe just a couple of things to add that we were discussing this yesterday. So we don't have a real view of the effect of climate change yet. So to do that, I think we're actually doing this. In a sense, we are characterizing the microbial communities over time. We don't have enough time points, let's say, to see what's already happening. Microbes usually can adapt pretty quickly to conditions. Although this will definitely have a change in the composition of the community. And there are some line of research actually showing that the climate change is already contributing to the emergence of novel fungal pathogens, including yeast. So as Jason mentioned before, there are many species of yeast also pathogens, and this seems to be fueled by the global climate change. And just to say that there is for storing the microbe itself, the yeast or bacteria and so on, is quite easy in a sense if you put in the right conditions. So we'll need to have some glycerol-based media. You can store in like -80 temperature, very cold temperature, almost indefinitely, and there are other methods to store cells, including, for example, desiccating them. So there are definitely methods like you mentioned for the seeds that enable us to keep almost indefinitely our collections.
Any other questions?
Thank you so much. So it's the second time I'm hearing your scientific stories, and I love them because it's brilliant, and it's also about cheese, wine, and walk in the forest. So thing that we all love. I have a question regarding human interaction because when we are not in your field, we tend to think that there is natural environment on the one hand, and then there is human action and human life on the other hand. Mm-hmm. And what is very interesting is that you have the question of human interaction with nature, which is not only about destruction, there is also a natural interaction. Yeah. So how does it work, and what is the loop, the human interaction coming back on the wild environment, and how this is changing or not the organism that you're studying?
Yeah. Yes, absolutely. So basically this human interaction for the microbes in a way it started involuntarily, so it started before actually humans they were aware of the microbes' existence and many of the notions we have it now. But it's incredible to see the impact that human had in the microbes. So it changed the position across the geography, it changed their genomes like for specific characteristic, and you can see specific signature in the genome that are different for each of the products that we use them. As you say, also there is this continuous flow from nature, let's say, to domestic, and from domestic to nature. So one thing that I didn't show you, but when we did all this sampling across Europe, we do find Saccharomyces cerevisiae in the forest, so we sample national park, national forest, and so on. We do see mostly around the Mediterranean area, in the south part of Europe and in North Africa. And then also we did see a few isolates in the very north of Europe, mostly in Sweden. But these were forest, a tree from. But when we sequence the genome, we do see actually these strains were isolated in different places. They actually turn out to be wine strains or beer strains that have been recently released to the wild. And the same things we did see with the olive oil strains. We do see, if we go to Crete, mostly to Greece, we did see plants, oak trees in the forest with this genotype. And we've been told after that actually it was common practice to throw away this olive oil wastewater after the extraction of the oil into the forest, basically. And that's where things are coming in and out, and they can recolonize natural environment, and in a way they can reshape this microbial community.
Yeah, I mean, it's definitely very interesting with, I think you had a great question, and this is pretty much, kind of the things we've constantly discussing in our research. I guess I have nothing more to add, but except of a comment that, because we are working with the genomes, and any of the changes throughout all these events or interactions can be traced, to a particular origin or particular reason or most likely scenario, which is nice because you do see, for instance, the Europe has, a tremendous amount of human history that influences the microbe itself. How the ale become lager and going to different places, and this is actually all can be found in the genomes, as well as as Gianni was saying, then if some of the strains escaped from the human environment to the natural surroundings, we can detect that, and we can actually see what then has changed once it's escaped from human civilization, human environment to the forest. Is there any additional change? And then even backwards, right? So this is the things we are constantly following, and it's very, very interesting. Yeah.
Thank you. So is there another question again? So if not, do you want to maybe develop or add something on an aspect of your presentation that you couldn't develop before?
Yeah, basically, maybe one thing to say that maybe it's a question of many is also how, for example, AI is it can be helping our research. So this is one thing that is quite embraced at the moment by science. So there is many applications driven by AI, for example, to study genomes, to, for example, try to predict how this genetic variation will impact traits. For example, we've been using AI to try to predict the traits themselves based on the genome sequence, so that could also help, let's say, to monitor this effect of, for example, the climate change. And more recently on this study where we sampled sites across Europe, because we have the history of the strains, we know where they came from, and actually from having the geographic position where they've been isolated, you can obtain climatic data on each of the sites, and we are currently trying to predict the distribution of the species. So even if we haven't done what you, for example, were suggesting about the global warming, we could try to predict in which environments our strains can be found, or at least could colonize based on their, let's say, on the information we had on their origin where they've been isolated. So, yeah.
Anything to okay, so probably one thing that I haven't really talked about is we have been working on, but this is only starting, is there's a lot of histories of fermentation in Asia, right? Not just like the typical baijiu or gaoliang that you probably observe in the Chinese culture. In Southeast Asia, in India, there's a lot of aboriginal fermentation practices, and it would be nice to relate some of the histories, and actually discover some of the yeast that's influenced by this aboriginal fermentation practices, and to see how the genetics changes were shaped by these changes. And I think this is something that you can actually relate to the culture and to the history as well as to the microbe. And this is something we like to do. Yeah.
Thank you. So, is there another question, or we stop there? Okay, so we'll stop there, but you will be able to continue the discussions one by one around the buffet if you want. Thank you so much to both of you for your presentations and everyone for the exchanges. I believe that thanks to these presentations and exchanges, we have learned a lot on yeast discoveries in population genomics and system biology. So thanks again to all of you for coming, and yes, I invite you to continue the discussions around bubble tea. Thank you.
Thank you. Thank you very much. It's really a pleasure. Hi.
你们真的有找到原住民上面有哪些项目菌种或是独特的用法？
有，就是应该是说，他们原住民后来再重新，就是因为之前是有公卖局，就是不准吃那样子的，所以其实有很多
祖传秘方就这样没了。
对，其实有很多 recipe 哈，就是那种其实是没的，但是后来现在其实也是在 COVID 之后，就是COVID 中间就是有些年轻人